Is going over our introduction to hematology PowerPoint. I recommend not watching the video while doing this. This is more like auditory flashcards. The questions are simple, the answers are short. But of course, as you're listening, the more you know, the more you can add. I will pause in between each question to hopefully give enough time to answer so you don't have to pause and restart the video every time. So let's go ahead and get started. What is hematology? The study of formed elements in the blood. What are 3 cellular blood elements? Red blood cells, white blood cells, platelets. What is hemostasis? Blood clot formation. What is hematopathology? Disease diagnosis via blood. Why is hematology important? It can detect disease early. What diseases can blood changes indicate? I'm looking for 3 answers here. Infection, anemia, and leukemia. Who first observed blood cells? Leeuwenhoek. When were platelets first described? 1800s. Who developed Wright stain? James Homer Wright. On to blood composition. What is average adult blood volume? 4.5 liters. Blood represents what percentage of body weight? 8%. What percentage of blood is plasma? 55%. What percent of blood is cells? 45%. What cell type is 99% of blood cells? Red blood cells. So the red blood cells account for 99% of the cellular components. What percent of cells are white blood cells? White blood cells make up 1%. And what percentage are platelets? Platelets make up less than 1% of cellular elements. Go ahead and name the 5 white blood cell types. Neutrophils, lymphocytes, monocytes, eosinophils, and basophils. Cellular elements. What is the function of red blood cells? To transport oxygen and carbon dioxide. What is the normal red blood cell shape? A biconcave disc. What is the normal red blood cell size? So on the screen here, if you've looked at it, it says 7 to 8 micrometers. But as we got further along in our PowerPoints and lessons, it actually opens it up more to like 6 to 8 micrometers is the normal red blood cell size. What are the red blood cell staining characteristics? So I'm looking at the color and then what is kind of interesting that happens in the middle. So they're salmon pink and they have a central pallor. And that central pallor takes up about 1/3 in a normal red blood cell. What are functions of the white blood cells? Immune defense. What are function of platelets? Primary hemostasis. What is the normal platelet size? 2 micrometers. So notice that is smaller than a red blood cell. Which white blood cell can diapedese? So that means it leaves the blood cell or the blood vessel and can actually go into tissue to fight infection. And those are neutrophils. Hematology testing. What is CBC? Complete blood count. What does a differential measure? White blood cell types in quantity. It's percentages. What is a peripheral blood smear? It's a microscopic morphology exam. What does ESR measure? Red blood cell sedimentation rate. What is the purpose of the ESR? It's a nonspecific inflammation indicator. CBC components. We're going to skip those because it didn't quite get it right, but I know we'll go back over them here in a different PowerPoint. What stain is used for smear? A blood smear. So it can be the right or the right Gemza. Right Gemza is typically for bone marrow. Which test counts immature red blood cells? The reticulocyte count. What examines bone marrow? This is kind of a silly one. Bone marrow exam. What are 3 tests for sickle cell? Solubility, electrophoresis, HPLC. What is the test for platelet function? A platelet function assay. And we use the light blue top tube, sodium citrate, for that, for the collection. For most other tests, we're actually going to be collecting in the violet tube top, EDTA. What's the primary role of the hematology technologists? Report data. Who do hematology technologists collaborate with, Hematopathologists. And what kind of problems do hematology techs need to solve? Problems with equipment and specimens. All right, for specimen collection, where is the blood collection site? Peripheral vein. It can also be done like straight to the heart. Hold on, we can look that up. Hold, please. Yeah, so peripheral stick or catheter, so going right into the heart there. Get back to where we were. Tube color for CBC, lavender. We just went over this. What's the lavender tube anticoagulant? EDTA. What is the EDTA mechanism? It chelates calcium. How long is the blood collected in a lavender top tube valid for at room temperature? 24 hours. What are the 2 forms of EDTA, and which one is preferred for CBC analyzers? So the 2 forms are tripotassium, which is liquid, and dipotassium, which is sprayed form. And this is the dipotassium EDTA is what's recommended for CBC analyzers. And why do we invert the tube after blood collection? Immediately after, we want to mix the anticoagulant. All right, cell structure and function. What are the 3 basic cell parts? Or 3 of them. I mean, we know that there's lots of cell parts. Membrane, nucleus, organelles. What are 3 membrane functions? Barrier, communication, interaction. What 3 components make up the membrane? Lipid, protein, carbohydrate. What's the nucleus function? Control center. Nucleolus function, rRNA synthesis. What are our chromatin types? Euchromatin and heterochromatin. And remember, euchromatin is loosely coiled and active and stains light. Heterochromatin is densely packed, stains dark, and not transcriptionally active. What is cytoplasm? It's the cytosol matrix. It's the function of ribosomes. Protein synthesis. What is the function of the Golgi? To modify and package macromolecules. What are the 2 different endoplasmic reticulum types? Smooth and rough. What is mitochondria function? ATP production. What's the function of the lysosomes? Digestive enzymes. What is the function of microfilaments? They are cytoskeleton support. What's the function of microtubules? Cell shape and mitosis. And what is the function of the centriole? It is the mitotic spindle anchor. All right, we're almost there. Cell proliferation and death. What do all blood cells arise from? Hematopoietic stem cells. What is it called when there are too few red blood cells? Anemia. What is it called when there are too many red blood cells? Polycythemia. What is it called when there are too few white blood cells? Immunodeficiency. What is it called when there are too many white blood cells? Malignancy. What are the 2 cell death types that can take place? Apoptosis and necrosis. What is apoptosis? It's programmed cell death. What is necrosis? Unplanned cell death. What are 3 triggers for apoptosis? Normal development, stress, or damage. What are 2 necrosis triggers? Hypoxia or trauma. Give me the 4 steps of necrosis. So with necrosis, the cell is damaged first. The membrane cation flux is lost. So we retain sodium in the cell. The cell swells because water follows the sodium. The membrane ruptures, and this causes inflammation in the body. How can we measure cell proliferation in the lab? With a Ki-67 assay. Apop, apoptotic, oh my gosh, apoptotic cells in hematology are called pycnotic nuclei.